Dear students, happy to meet you in this online mode of education. The topic I am going to discuss with you today is about the crossing over, which is coming under the paper genetics. I am Dr.S.ANGELINA, PARIMALA, Associate Professor and Head from the PG and Research Department of Zoology, A.D.M.COLLEGE for Women, NAGAPATTINAM. Let us see something about the introductory part of crossing over. Let us see what is crossing over. It is a process which occurs during meiosis cell division, where homologous chromosomes exchange segments of their genetic material. This results in genetic recombination and contributes to genetic diversity. Actually, this crossing over is a basic concept of genetics and cell biology, and this is often called as recombination. It is the exchange of chromosome segments between non-sister chromatids during the production of gametes. The effect is to assort the alleles on parental chromosomes so that the gametes carry combinations of genes different from either parent. This has the overall effect of increasing the variety of phenotypes present in a population. The process can be seen directly in stem cells and indirectly if there are genetic markers or not on the chromosomes. The visible crossovers are called chiasma. In plural, they are called as chiasmata. The large scale effect of crossing over is to spread variation through a population. This is the main result of sexual reproduction compared to non-sexual mode of reproduction. The main advantage to the parents is a greater variety in their offspring. Thus, it is thought increases the chance that some offspring will survive and reproduce. Let us see something about the process of crossing over. So, it is taking place in the meiosis cell division at the time of meiosis 1. During prophase 1 of this phase, here homologous chromosomes, that is each consisting of two sister chromatids, pair up in a process that is called synapsis. Next is the formation of chiasma. Here the homologous chromosomes exchange segments at points, that is called chiasma. Let us see something about the exchange of genetic material. The exchange of genetic material between non-sister chromatids results in recombinant chromosomes which have a mix of alleles from both parents. Let us see something about the types of crossing over. Here it was classified into single crossing over, double crossing over and multiple crossing over. Let us see something about the single crossing over. Here it involves one chiasma and results in the exchange of genetic material between two chromatids. The next is about the double crossing over. Here it involves two chiasmata. This can be result in a segment of the one chromatid being exchanged twice, which can produce complex recombinant patterns. The next one is multiple crossing over. Here it involves more than two chiasmata, and this crossing overs can occur along the length of homologous chromosomes, increasing genetic variation. Some examples we can see related to crossing over. Example 1 is about the Drosophila melanogaster. This is commonly called as fruit fly. Here we can consider genes A, B and C located on the same chromosome. If crossing over occurs between genes A and B, the resulting gametes might show new combinations of these genes. If the original chromosome has alleles A, B, C and its homologue has a small ABC, a crossover between capital A and capital B can produce new combinations like capital A, small b and capital C, or small a, capital B and capital C. Let us see something about the importance of crossing over. When we see, the first one is about genetic diversity. Here, this crossing over increases genetic variation in populations by creating new combinations of alleles. This diversity is crucial for evolution and adaptation to changing environments. The next one is about inheritance patterns. It helps explain the inheritance of linked genes and why offspring may show new combinations of traits not seen in parents. The next is about genetic mapping. Here the crossing over frequencies are used to construct genetic maps. The frequency of recombination between genes is proportional to their distance on a chromosome, allowing scientists to map the relative position of genes. The next is about biological research. Here we can see understanding crossing over is fundamental in genetic research, helping to elucidate the mechanisms of genetic disorders and the function of genes. The last one is about the breeding program here in agriculture and animal breeding. This control crossing over can be used to combine desirable traits from different strains of breeds. Let us see something about the molecular mechanisms of crossing over. Here, formation of synaptonymal complex. Here, during synapsis, the synaptonymal complex forms between homologous chromosomes, facilitating the pairing and the exchange process. The next one is about double strand breaks. Here, this process is initiated by programmed double strand breaks in the DNA. Enzyme structures SPO11 in yeast catalyze these breaks. Homologous recombination pairs. Here, it breaks and required using the homologous chromosome as a template, leading to the exchange of genetic material. Then, resolution of holiday junctions. The cross-shaped structures known as holiday junctions are resolved, resulting in separation of homologous chromosomes with exchanged segments. And also crossing over occurs during prophase 1 of meiosis in a subphase called packaging. Here, step by step outline process we can see. First one is about the formation of the synaptonymal complex. Here, the homologous chromosomes pass up along their length, forming a structure called the synaptonymal complex. This pairing process is known as the synapsis. The next one is double-stranded break formation. Here, the involvement of enzymes such as SPO11 in yeast induce double-strand breaks in the DNA of one of the homologous chromosomes. Let us see something about strand invasion. Here, the broken DNA ends are processed to create single-stranded overhangs. These overhangs invade the corresponding sequences on the homologous chromosome, forming a structure called as zero. The next about the holiday junction formation. Here, the invading strand pairs with the complementary strand on the homologous chromosome, displacing the original complementary strand. This leads to the formation of cross-shaped structures known as holiday junctions. Next is about branch migration. The holiday junction strand migrate along the DNA, extending the region of heteroduplex. The next is resolution of holiday junctions. Here, the junctions are resolved by specific endonucleases, resulting in the exchange of genetic material between homologous chromosomes. The resolution can produce two outcomes, either crossover products or non-crossover products. Let us see something about the theories of crossing over. There are many theories have proposed to explain the mechanics and the evolution of crossing over. The best one is classical theory, that is otherwise called as chiasma theory. It is proposed by Franz Hans Jacens in 1909. This theory suggested that crossing over is initiated at visible points of contact between homologous chromosomes called chiasmata. The exchange of genetic material occurs at these chiasmata during meiosis. The next one is Stern's model. Here, Stern's studies in Drosophila melanogaster provided evidence that crossing over involves the physical exchange of chromosome segments. His work demonstrated that crossing over results from the breakage and reunion of chromosome arms. The next theory is the breakage and reunion theory. It is proposed by Harry Stevenson and Barbara McIntosh in 1930. They provided psychological evidence for crossing over by demonstrating that genetic recombination is accompanied by the physical exchange of chromosomal segments. The next one is the double strand break repair model. It is proposed by Jacob and his colleagues in 1983. This model explains that crossing over is initiated by double strand breaks in the DNA. The repair of these breaks through homologous recombination involves strand invasion, formation of holiday junctions, and their resolution. The next model is the Michelson-Rading model. Here, it is an alternative to the double strand break model proposed by Matthew Michelson and Shalon Rading. This model suggests that recombination is initiated by a single strand break in one DNA molecule, followed by strand invasion and the formation of a heteroduplex. The next theory is about the synthesis-dependent strand annealing model. This model suggests that recombination can occur without the formation of Holiday junction. After the strand invasion and DNA synthesis, the invading strand dissociates and anneals to the other end of the brain, leading to gene conversion without crossing over. Then let us see something about chromosomal crossover. Here it is the exchange of genetic material during sexual reproduction between two homologous chromosomes known as the chromatids that results in recombinant chromosomes. It is one of the final phases of genetic recombination which occurs in the packaging stage of phase 1 of meiosis during the process called synapsis. Here it begins before the synaptonymal complex develops and is not completed until near the end of phase 1. Crossover usually occurs when matching regions on matching chromosomes break and then reconnect to the other chromosome. And also it was described in the theory by Thomas and Morgan. The term crossover was coined by Morgan and Elliott et al. And relied on the discovery of Franz Alphonse Dassens to describe the phenomenon in 1909 and had called it chiasmatitis. The term chiasma is linked, if not identical to crossover. Morgan immediately saw the great importance of Janssen's psychological interpretation of chiasmata to the experimental results of his research on the heredity of Drosophila. The physical basis of crossover was first demonstrated by Harris Crichton and Barbara McClintock in 1931. The linked frequency of crossing over between two genes loci is the crossover value for a fixed set of genetic and environmental conditions. Recombinations in a particular region of the linkage structure tends to be constant, and the same is then true for the crossing over value which is used in the production of genetic maps. When Hota et al. In 1977 compared meiotic crossing over in lily and mouse, they concluded that diverse eukaryotes share a common pattern. This finding suggested that chromosomal crossing over is a general characteristic of eukaryotic meiosis. When we see the origin, there are two popular and overlapping theories that explain the origin of crossing over, coming from the different theories on the origin of meiosis. The first theory explains about the idea that meiosis evolved as another method of DNA repair, and thus crossing over is a novel way to replace possibly damaged sections of DNA. The second theory comes from the idea that meiosis evolved from bacterial transformation with the function of propagating diversity. Let us see something about DNA repair theory. Here, crossing over and DNA repair are very similar processes which utilizes many of the same protein complexes. The report says that the significance of responses of the genome to challenge. Here we can see the genome would change itself to overcome threats to its survival. And here the scientists used to explain with 450 self-pollinated plants that received from each parent a chromosome with a repeated end. And here also we can see the modified patterns of gene expression on different sectors of leaves of the corn plants that shows the transferable elements hide in the genome, and their mobility allows them to alter the action of genes at different loci. This evidence can also restructure the genome anywhere from a new nucleotides with the whole segments of chromosomes. The recombinases and primases lay a foundation of nucleotides along the DNA sequence. One such particular protein complex that is conserved between processes in ORGAD51, a well-conserved recombinase protein that has been shown to be a crucial DNA repair as well as crossover. Several other genes in Drosophila melanogaster have been linked as well to both processes by showing the mutants at these specified loci it cannot undergo DNA repair or crossover. DNA repair and crossover have been found to favor similar regions on chromosomes. Furthermore, crossing over has been correlated to occur in response to stressful and likely DNA damaging conditions. When we see the link to bacterial transformation, here the process of bacterial transformation also shares many similarities with chromosomal crossover, particularly in the formation of overhangs on the sides of the broken DNA strand, allowing for the annealing of a new strand. Bacterial transformation itself has been linked to DNA repair many times. The second theory comes from the idea that meiosis evolved from bacterial transformation with the function of propagating genetic diversity. Thus, its evidence suggests that it's a question of whether crossover is linked to DNA repair or bacterial transformation. As the two do not appear to be mutually exclusive, it is likely that crossover may have evolved from bacterial transformation, which in turn developed from DNA repair, thus explaining the link between all three processes. A current model of meiosis recombination initiated by a double-strand breakup or gap, here followed by a pairing with a homologous chromosome and strand invasion to initiate the recombinational repair process. Repair of the gap can lead to crossing over or non-crossing over of the flanking region. Crossing over recombination is thought to occur by a double Holiday junction model, which is illustrated on the in the recombination model, and also here they are thought to occur primarily by a synthesis dependent strand annealing model, and also we can see the recombination events appear to be in the HDSA type. And also meiotic recombination may be initiated by double-stranded breaks that are introduced into the DNA by exposure to DNA damaging agents or the SPO11 protein. One or more exonucleases then digest the 5' ends generated by the double-stranded breaks to produce 3' single-stranded DNA tails. The meiosis-specific recombinase DMC1 and the general recombinase RAD51 coats the single-stranded DNA to form nucleoprotein filaments. The recombinase catalyzes invasion of the opposite chromatid by the single-stranded DNA from one end of the break. Next, the 3' end of the invading DNA primes DNA synthesis, causing displacement of the complementary strand, which subsequently anneals to the single-stranded DNA generated from the other end of the initial double-stranded break. The structure that results is a cross-strand exchange, also known as a Holiday junction. Here, the contact between two chromatids that will soon undergo crossing over is known as the chiasma. The Holiday junction is a tetrahedral structure which can be pulled by other recombinases, moving it along the four-stranded structure. This is something about particularly the importance of chiasma. In the grasshopper melanopus, here we can, when it was exposed to an acute dose of X-rays during each individual stage of meiosis, and the chiasma frequency was measured. The radiation during the lecithin zygotine stages of meiosis was found to increase the subsequent chiasma frequency. Similarly, in the grasshopper, here it was exposed to radiation during the zygotine stage. Here, the early packaging stages caused a significant increase in mean cell chiasma frequency, and also it was stored at the later diplotin diagenesis stages of meiosis. This result suggests that X-rays induce DNA damages that are repaired by a crossover pathway leading to the chiasma formation. This is something about the difference between gene conversion and chromosomal crossover. In most eukaryotes, a cell carries two versions of each gene, each referred to as an allele. Each parent passes on one allele to each offspring, and individual gametes inherit a complete haploid complement of alleles on chromosomes that are independently selected from each pair of chromatids lined up on the metabase plate. Without recombination, all the alleles for those genes linked together on the same chromosome would be inherited together. Meiotic recombination allows a more independent segregation between two alleles that occupy the positions of single genes as the recombination shuffles the allele content between homologous chromosomes. Recombination results in a new arrangement of maternal and paternal alleles on the same chromosome. Although the same genes appear in the same order, some alleles are different. In this way, it is theoretically possible to have any combination of parental alleles in an offspring, and the fact that two alleles appear together in one offspring does not have any influence on the statistical probability that another offspring will have the same combination. This principle of independent assortment of genes is a fundamental to genetic inheritance. However, the frequency of recombination is actually not the same for all gene combinations. This led to the notion of genetic distance, which is a measure of the recombination frequency averaged over a sample of pedigree. Here, one may say that this is because recombination is greatly influenced by the proximity of one gene to another. If two genes are located close together on a chromosome, the likelihood that a recombination event will separate these two genes is less than if they were far apart. Genetic linkage describes the tendency of genes to be inherited together as a result of their location on the chromosome. Linkage disequilibrium describes a situation in which some combinations of genes or genetic markers occur more or less frequently in a population that can be expected from their distances apart. The concept is applied when searching for a gene that may cause a particular disease. This is done by comparing the occurrence of a specific DNA sequence with the appearance of a disease. When a high correlation between the two is found, it is likely that the appropriate gene sequence is really closer. Let us say something about non-homologous crossover. Here, the crossover typically occurs between homologous regions of matching chromosomes, but similarities in sequence and other factors can result in mismatched alignment. Most DNA is composed of base pair sequences repeated very large numbers of times. These repetition segments, often referred to as satellites, are fairly homogeneous among a species. During DNA replication, each strand of DNA is used as a template for the creation of new strands using a particularly conserved mechanism. And also proper functioning of this process results in two identical paired chromosomes, often called sister chromatids. And also these sister chromatid crossover events are known to occur at a rate of several crossover events per cell division in eukaryotes. Most of these events involve an exchange of equal amounts of genetic information, but unequal exchanges may occur due to sequence mismatch. These are referred to by a variety of names, including non-homologous crossover, unequal crossover, and unbalanced recombination. And this results in an insertion or deletion of genetic information into a chromosome. While rare compared to homologous crossover events, these mutations are drastic, affecting many loci at the same time. They are considered the main driver behind the generation of gene replication and as general source of mutation within the genome. The specific causes of non-homologous crossover events are unknown, but several influential factors are known to increase the likelihood of an unequal crossover. One common vector leading to unbalanced recombination is the repair of double-strand breaks. Here we can often repair using homology-directed repair, a process which involves invasion of a template strand by the DNA strand. And also nearby homologous regions of the template strands are often used for repair, which can give rise to either insertion or deletion in the genome if a non-homologous but complementary part of the template strand is used. And also sequence similarity is a major player in crossover, and also in the crossover events are more likely to occur in long regions of close identity on a gene. This means that any section of the genome with long sections of repetitive DNA is prone to crossover events. The presence of transferable elements is another influential element of non-homologous crossover. The repetitive regions of code characterize transferable elements, complementary but non-homologous regions are ubiquitous without gene transfer. And also because of chromosomal regions composed of transforms have large quantities of identical repetitious code in a condensed space. It is thought that transcription regions undergoing a crossover event are more prone to erroneous complementary match up. And also, this can say that a section of a chromosome containing a lot of identical sequences should undergo a crossover event is less certain to match up with a perfectly homologous section of complementary code and more prone to binding with a section of code on a slightly different part of the chromosome. This results in unbalanced recombination as genetic information may be either inserted or deleted into the new chromosome depending on where the recombination occurs. Let us see something about the significance of crossing over. When we see here, the crossing over produces a new combination of genes, and also crossing over plays an essential role in the process of evolution. And also crossover frequently helps in the construction of genetic maps, and also crossing over provides evidence for a linear arrangement of linked genes in a chromosome. And also the crossing over provides an inexhaustible store of gene variety in a sexually reproducing organism. And also crossing over refers to the exchange of genetic material or chromosome segments between non-sister chromatids in meiosis. This genetic process occurs between homologous regions of matching chromosomes and the interchange of homologous chromosomes. And also crossing over is an enzyme-mediated process where the exchange of genetic material between non-sister chromatids of homologous chromosomes takes place. It occurs at the packaging stage of process 1 of meiosis. And also crossing over is a vital process in meiosis that promotes genetic diversity by recombining genetic material between homologous chromosomes. Understanding its type, mechanisms, and significance is essential in fields ranging from evolutionary biology and genetics to agriculture and medicine. The study of crossing over not only elucidates the principles of inheritance but also provides valuable insights into genetic mapping and the underlying causes of genetic disorders. Let us conclude the importance of crossing over here. It is a cellular process that happens during meiosis cell division, and this mechanism of crossing over involves a complex series of events starting with the formation of double strand breaks, followed by strand invasion, formation of colgate junctions, and their resolution. And also theories such as the classical chiasma theory, breakage and reunion theory, and the double strand break repair model, they provide insights into the process of underlying genetic recombination. This process is crucial for generating genetic diversity, accurate chromosome segregation, and constructing genetic maps with significant implications for biology, medicine, and evolution. Thank you.